Lori, and the title of my talk is Age-Related Differences in Emotional fMRI Responses After Accounting for Cerebrovascular Reactivity Decline. So the heart of my study is about emotional aging. And what that means is that across the adult lifespan, we know that many cognitive functions actually decline, such as the speed of processing, working memory function, and also reasoning. But on the other hand, with aging, our emotional regulation function remains stable, and actually, in some aspects, it actually improves. So, for example, we see that elderly will respond less negatively when presented with a negative stimulus, and they can also restore their mood more effectively after negative mood manipulations. So, previous fMRI findings on this topic have used bold fMRI, and they reported age-related increases in the activation of the prefrontal cortex. But on the other hand, there were inconsistent findings in the amygdala. So some studies reported age-related decreases in amygdala activity, others saw increases, and some saw age-invariant activity. And maybe that is because of the use of the bold fMRI signal itself. So as we are all probably very aware, bold is an indirect measurement of neuronal activity. So the mechanism of it is that given a stimulus, it triggers neuronal activity that through neurovascular coupling results in a hemodynamic response that we can then detect with our MRI scanner and get an fMRI bold signal and get our bold images. So I want to focus on this step here. So the hemodynamic response is based on blood flow, blood oxygenation, and those types of things. But with normal aging, we experience cerebrovascular changes. And that includes that the arterial walls lose elasticity and there's arterial stiffening. And this results in changes in blood flow. And we also see tortuosity of vessels, which is that twisting, and that affects the perfusion. And then we also see that there are changes in the neuroglial vascular signaling, and this leads to impaired cerebrovascular reactivity with age. And these are all results of normal aging. So aim of this study is to characterize the age-related differences in the emotional circuit through fMRI signal in the context of vascular function using CVR. And this will help us to determine if previously reported age-related changes are due to decline in vascular function rather than diminishing neural activity. And we will do this by comparing activity in the amygdala, the left inferior frontal gyrus, and the right inferior frontal gyrus before and after accounting for vascular factors. So our study included a 10-minute task-evoked fMRI scan and also a 7-minute hypercapnia inhalation MRI. And we recruited 27 healthy subjects that we separated into two groups, a young group consisting of 16 participants aging from 23 to 38, and the older group consisting of 11 participants with an age range from 53 to 73 years old. And our emotion task fMRI used this block diagram in the corner here, where we had 30-second fixation periods and 30-second image viewing periods. And in the 30-second image viewing periods, we showed the subjects 10 emotional pictures, positive images, neutral images, and negative images. And for our hypercapnia inhalation MRI, we had 70-second blocks of 5% CO2 breathing, followed by 50 seconds of room air breathing, and we had 3 CO2 breathing blocks. And we also had some psychiatric measures. So that was questionnaires that we gave to the participants to measure their moods and their emotions. And those moods were positive affectivity, cognitive reappraisal, agreeableness, extroversion, and stress. So positive affectivity is like someone's ability to look on the bright side of things, and cognitive reappraisal is the ability to overcome a negative event. So for our data analysis, our CVR maps were obtained using standard GLM methods, and our CVR images were co-registered to the task fMRI bold images and then normalized into M&I space. It's important so that we can directly compare our ROIs. And we use 3 picture contrasts. So that was positive versus neutral images, negative versus neutral, and negative versus positive images. And the psychiatric mood measures were also compared between the young and old groups, and their associations with the bold responses were evaluated before and after CVR correction. And these are some examples of the ROI regions that we use, the amygdala, left IFG, and right IFG. So starting with our bold results, in the upper corner here, we have our group activation map, which shows that we had activation in the left IFG, right IFG, and amygdala. So that's sort of a proof of concept that these regions are activated. And if we look at our bold results for each of our contrasts at the bottom here, we can see that there was no significant age difference in the amygdala for any of the contrasts. But in the left IFG, we saw that the older group had significantly higher fMRI signal for all 3 of our contrasts. While in the right IFG, we only saw a trend of significance in the negative versus positive contrast, with the older group generally having higher fMRI signal and activation. And then for our CVR results, we have our group average CVR maps. And visually, we can see that the young group has a brighter overall CVR, which corresponds with our knowledge that the older group has a lower cerebrovascular reactivity, and that can sort of easily be seen, like looking at these regions here, the young group is much brighter. So we also can see that sort of decrease in cerebrovascular reactivity, looking here at our 3 ROIs. And in all cases, the young group has higher CVR compared to the older group. And in the prefrontal regions, that difference between the 2 groups is significant. And when we further break down these ROIs into their image contrasts, we saw in the negative versus positive contrast in the amygdala that there was a slight significance with the younger group having higher CVR. And in the left IFG, we saw significance in all 3 of our contrasts, that the younger group had higher CVR. And in the right IFG, we didn't see any significance, but we can still see that the younger group had some slightly higher CVR. And so earlier when I talked about how we were going to correct for the change in vascular with age, you're probably wondering how we would do that. And it's a very simple formula. So all that we do is we take our task bold fMRI signal and we divide it by our CVR. And we do that for each subject. And that gives us the corrected bold over CVR. And we can see with these results that after correction and before correction, there was no age difference in the amygdala. But in the left IFG, there was an age difference in all three of our contrasts. And after we did our CVR correction, that age difference actually became more significant. And we also saw this in the right IFG, that when it was uncorrected, we only saw a slight significance in the negative versus positive contrast. But after correcting for the cerebrovasculature, we saw that trend of significance in all three of our contrasts. We also looked at some correlation coefficients between our psychiatric measures and fMRI signal. And the green boxes are the significant boxes. And what we saw in the amygdala was that after correction, we no longer had any significance in positive affectivity, but we saw a new significance in cognitive reappraisal. And then we also lost some significance in stress in the amygdala. And what's interesting about this is with our corrected results, we see that cognitive reappraisal has a negative correlation and stress has positive correlations. And this sort of aligns with our knowledge of the amygdala being the fear center of the brain. So we have that positive correlation with stress, which is a negative emotion. And then we have a negative correlation with cognitive reappraisal, which is a positive emotion. And then in our prefrontal regions, we see some increased significance after we do our correction. And we see that in both the left and the right IFG. And the IFGs are associated with just emotion processing. So that's why we see they have positive correlations with stress and agreeableness, extroversion, all those positive and negative moods. So a brief summary of our findings. So our bold fMRI findings are consistent with previous literature. And we found a general decrease in CVR between the young and the old groups. And after accounting for CVR changes, the bold signal saw no change in amygdala activation, but we saw greater prefrontal activation in the older group. And with our psychiatric measures, we saw greater correlation in the prefrontal regions after CVR correction and less correlation in the amygdala after correction. So our fMRI findings and our psychiatric measures sort of go together in that way. And for a discussion, we saw age invariant activity in the amygdala, and that kind of shows that the function of the amygdala remains intact in emotional aging. And the older group had significantly lower CVR measurements in the left and right IFGs compared to the younger group, yet there was higher bold activation. So that confirms that shift towards prefrontal activation in aging. And the significant age-related increase in prefrontal activity is thought to represent a compensatory activation to accommodate decreased volume and efficiency of neural circuits. And finally, accounting for the brain vasculature can give more accurate insight into the fMRI responses that we measure. And this study provides the foundation for future studies regarding emotional circuitry and the contribution of vascular responses in normal aging.